In this video, we are going to talk about the endo-membrane system inside the cell and how a protein is made, packaged, and moved around the cell. So what is the endomembrane system? It's a collection of membrane-bound organelles that work together to modify, package, and transport lipids and proteins. So it involves the endoplasmic reticulum, both rough and smooth, these vesicles, the Golgi apparatus, and all of these that are involved in this function. So we see that the rough endoplasmic reticulum is involved in making protein, then shipping it to the Golgi apparatus, and then out of the Golgi apparatus, the modified protein comes out and it's shipped to a particular place in the cell. So let's follow closely as to how it happens. So in this example, we're looking at a membrane-bound protein. What we could be looking at, or this could be an example of a protein that is not membrane bound, but it is made and soluble in inside the endoplasmic reticulum, which is called the lumen of an endoplasmic reticulum. So the protein is made used on these ribosomes. The protein is, in this case, is a membrane bound protein. It's sitting in the membrane, and when it is ready, this vesicle buds out of the endoplasmic reticulum, moves to the Golgi apparatus, and this vesicle fuses with the cis face of the Golgi apparatus. Then this protein travels through the different layers of the Golgi apparatus. It folds, becomes modified, and then it is ready to be shipped out by the time it reaches the trans phase. And when it reaches the trans phase, it buds out as a transport vesicle and is transported to the plasma membrane. This transport vesicle fuses with the plasma membrane and then places the protein on the plasma membrane. If this was a secretory protein, this protein would be inside this space, inside this vesicle, and then as the vesicle would fuse with the plasma membrane, it would just spit out the protein outside the cell. And this happens in the cell all the time. For example, when your pancreatic cells create insulin and then they release it, that's how it happens. So now let's talk about the cytoskeleton and the extracellular matrix. And as the name implies, cytoskeleton is involved in giving the cell structural integrity. There are three different types of cytoskeletons, and they're named based on the thickness of these fibers. They're usually, these proteins are in the form of fibers, and they're all made of proteins. So we have microfilaments, which are the smallest thickness of these type of proteins. We have intermediate filaments, which, as the name implies, has an intermediate diameter, and then the microtubules are the thickest ones. The intermediate filaments, because it's an artist's interpretation of if you were to stain these proteins, that's what you would see inside a cell. So their main function is maintaining the shape of the cell and also help anchoring those various different organelles that is inside the cell in the cytoplasm. So it's not like things are just floating around aimlessly, they're anchored. Microfilaments are these filaments that is shown in green. They are involved in muscle contraction. A good example of it is actin molecule. And microfilaments are also involved in division of the cytoplasm when a cell divides. So when an animal cell divides, these microfilaments become really tense, and then they pinch the membrane of the cell and breaks it in half. You'll hear more about this when we talk about cell division. Microtubules are involved in a number of different things. One of them is cell movement. You've seen flagella before with bacteria, and we talked about the fact that this flagella is also used in sperm to help it move around. Cilia are kind of the same kind as the flagella, except they're shorter. An example of it is these short protrusions that come out of your air pathways in your throat, the pathways that moves towards your lungs, and these cilia move side to side. And when something, a particle, gets into your throat, or a virus particle or bacteria, they kind of move side to side and push those components out of the area up, so they prevent them to move down into your lungs. Microtubules play a very, very central and important role in cell division as well. They help the cell divide the genetic material into half when it divides, so that when you make two new cells, both ends of the cell, both ends that come out of the cell division, have the same amount and kind of genetic material. And you can see them stained in here. And these are really huge fibers of proteins, really long, that are made from structures called centrioles. Each cell, each animal cell, has a couple of centrioles. And from this, these long microtubules are formed and get extended. You will see more of these microtubule functions and hear about centrioles when we talk about cell division. Microtubules also guide organelle movement. They kind of form highways on which vesicles can move. So remember when we were talking about here, when we were talking about these vesicles moving inside the cytoplasm, they're not really floating aimlessly. They're actually moving along highways that are made of these cytoskeleton components. So what happens, if you take a look at this image, this huge ball is a vesicle that carries something. And then these are either microtubules or intermediate filaments. These are the highways on which these vesicles move. And what allows them to move are these motor proteins that basically pull them on these highways. Now, this is a very interesting illustration, and I will show you. So as you can see, this motor protein is actually, and this is actually what's happening inside the cell. Using the energy of ATP, this protein keeps changing its shape, and it's just like a little person walking and pulling something. Now, we've been talking about components of a cell inside the cell, but there's actually lots happening outside of a cell. First of all, a lot of the cells in multicellular organisms actually are anchored to their place. And the reason that they can get anchored is because the cells are able to connect and anchor to proteins that are sitting in the what's called extracellular matrix. The extracellular matrix is an area outside of the cell. And these connections are made through the plasma membrane. And what aids in interaction are proteins, transmembrane proteins that sit across the membrane, and an accessory protein called fibronectin. Now, fibronectin allows this integrin proteins to connect with components of the extracellular matrix, such as the collagen fiber or these proteoglycan complexes. Proteoglycan complexes are these huge carbohydrate molecules that have little pieces of protein attached to them. These collagen fibers play a very important role in structural integrity of our body systems. As we age, these collagen fibers break down, and this is the reason that actually we get wrinkles. A function of these proteoglycan complexes is cell-to-cell interaction.